Okay, this is the prep for lecture 27. I had a phone call right in the middle, and so I have to start over, which is all right. We're going to finish up the neural tube sort of patterning. We'll talk about anterior posterior patterning a little bit, and just the very basics of the formation and patterning of the brain, with a little bit of information, with a small number of gene names to know. Both for the anterior posterior patterning of the brain, as well as the cells within the layers that form as the brain is developing. A little bit about the neural tube, and then we'll spend probably half of the time on neural crest, which is very interesting. It forms between the surface ectoderm and the neural tube. It is derived from ectoderm, but interestingly, some of the cells it ends up forming are mesodermal and some are ectodermal. And it migrates around and forms a huge variety of different topics. And I will probably, in the lecture, introduce a potential, again, optional, always optional, but a bonus question about neural crest that some students might find, I hope, quite interesting. Okay, there goes my neighbor, thump, thump, thump. So just remind you of the two signal model for anterior and posterior patterning of neuroectoderm. We've already kind of talked about um, this idea in general terms in previous classes. So I'll just basically recap that there are signals that are sent by the mesoderm to the ectoderm overlying the organizer region, so just above it, to induce neural tissue to develop. And this is TGF beta inhibitors, mostly BMP inhibitors, and these prevent BMP signaling, which is required to repress neural tissue formation. And then there's also, you will remember, Sonic Hedgehog comes from the notochord, and it basically it is patterning sort of the closest part of the neural plate, neural tissue, and there is going to be Sonic Hedgehog expressed from the notochord here too. So these are all the kinds of signals we've already talked about. And just to point out that the neural tube is not the same anterior to posterior. Of course, the most anterior part is going to form the brain, and then the more posterior region will form neural tube. And each region of the neural tube, although they look quite similar through, with the neurons coming in and neurons going out of the tube, innervating the body and receiving signals from sensory information, they're going to be innervating different parts of the body, so there's going to be some posterior anterior patterning taking place there. And it's thought that there is this Wnt signal, a graded signal from the mesoderm, where you have the strongest signal in the posterior and the weakest in the anterior. And we've already met Wnt signaling and shown how it gives this posterior to anterior mesoderm fate. And we've have already seen, or we will see at some point, that the mesoderm is basically that's below the ectoderm, can signal for the different fates, anterior versus posterior. So this is all just to say that we don't know all the details about this, but what the people that research this, I am not one of them, but it's known that Wnts are part of this process, and it is also thought that retinoic acid gradient of some kind is also taking part in this. And so it's not entirely understood. There's a lot of these processes, you'll notice when I tell you what is known about some of them, that there will be there have been and will continue to be places where I go, but we don't know all the details yet. This is how we think it happens. This is a model. So far it explains the evidence and so on. And so this is one of those kinds of cases. Okay, so as far as the anterior neural tube goes, whoop, you don't want that. We want a pen. The anterior part is going to form the brain, and the brain is quite spread out at first. I hope you can't hear this thumping. People next door. And it's divided into multiple sections, and the first divisions are just the forebrain, the midbrain, and the hindbrain. And there are all these little indentations that section off the different parts, and they're referred to as neuromeres to begin with. So each one of these divisions is a neuromere. And then later in brain development, I'll just point out where there are sort of slightly different names. But the prosencephalon is the name for the forebrain, mesencephalon for the midbrain, and rhombencephalon for the hindbrain. It's okay if you just remember these terms. That will be all right. And then in the posterior, as I said, we're talking about the spinal cord, but of course the different the places where the nerves that emanate from or are coming back from the body, the destinations for those are going to be different. So there's going to still be some different patterning along the anterior posterior axis. I am just gonna, because I've already done this one time, I'm just going to remove some of the stuff I put in there just so that I can add it as I talk. I do find that can be helpful. Okay, so one thing I tried to do with this diagram was to just color code a little bit in the description to match the information here. So if we look at the 2-day mouse embryo, we see 2 transcription factors that are being expressed. So the mid and forebrain have OTX2, and then GBOX2 is in the hindbrain. And just note that these are referred to as rhombomeres just during the time that the rhombencephalon, the hindbrain, is forming, because there's going to be a dilation of this region, and it's not going to be a whole bunch of separate sections once that is finished. So the R1 through R7, those are just a temporary designation of the different parts, and each of these is going to, contribute to different parts of the developing brain. Remember, there is sonic hedgehog from the notochord, and in the floor plate, the most ventral part of the neural tube developing. So this is constant throughout the sonic hedgehog. There's going to be an additional signal a little bit later. And what else was I going to tell? Okay, so I'll just mention the names now. So the anterior neural ridge is in the most anterior part of the developing forebrain, the prosencephalon, and it is a source of FGF signals. So it's sending signals to this region, and this is going to give rise to the forebrain, and it's not going to have any of the OTX expression even just a couple of days later. This is a 4.5-day embryo. Okay, so that's the role of that. And then the ZL1, zona limitans intrathalamica. And so that's, if you just remember ZL1, that's going to be fine. And it's not doing very much at this early stage, but in the 4.5-day embryo, it's starting to express sonic hedgehog to the cells to either side of it. And so it's part of the signaling, and it's basically helping the thalamus, which is part of the brain, and some prethalamus cells on the other side. So these, that's where the intrathalamica is what that means. And it's part of patterning the forebrain. And then there's the midbrain-hindbrain boundary, which is the only one really in some ways that is easy to remember because it tells you exactly where it is and what it's doing. It's a boundary between the hindbrain and the midbrain. And it is also sending FGF signals on either side here. And it's creating this very distinct boundary between the OTX and the GBOX expressing cells, so that the midbrain is forming a structure called the tectum. The hindbrain is developing the medulla and the cerebellum. And the cerebellum, you may remember, is for balance and sort of motor control. And I think that is really all I would like you to remember. I've tried to, to be careful with the color coding so that you can see the consistency. So these are the three sort of local organizers that you need to know. And basically what they're doing is they're specifying that the ANR is specifying the most anterior part of the forebrain, and the ZL1 is kind of the second part of the forebrain, which includes the thalamus, which is an important gland. And then the MHB is specifying the separation, the boundary distinctly between hindbrain and midbrain. And that's the main things for you to remember. So the brain is more than just an expanded tube. Of course, there's a lot of things going on in there. This is a early mouse embryo, and it's just a section through a developing brain. And the only purpose for these genes, this is SOX2, this is TBR2, and TBR1. And the only purpose for including them is that they just show you, like when the embryo is stained, the brain is stained for these are markers that help you determine the kinds of cells that are found there. And so the SOX2 is the neurogenic precursor, or neural stem cells, let's say NSC, neural stem cells. And so these are the cells that they can form pretty much any neural type of fate. So that means not just neurons, but some of the supporting cells that are not themselves sending messages, but are actually, they're providing nutrition and support and other sorts of functions. So, and if we look at the ventricle region, I've shown this picture before, but if you think of the ventricle as a big sort of cavity inside the brain, cells have apical and basal polarity, so there's different molecules often on one surface relative to the other. And in the case of these developing brain cells, the apical surface is facing into the cavity, and then the basal surface is away from that. So if these were all single cells, this would be the apical and this would be the basal surface. Okay, and if these are layers of cells, they're going to be apical basal and then apical basal like that. So that is going to be how they are organized. And so the SOX2, those, as I said, those are the ones that have neural cell fates. And then above those are progenitor cells. So neural stem cells divide to make more stem cells and more stem cells. And then those progenitor cells, when they develop slightly, have the ability now to form either neuronal or glial cell fates. And when they are ready to divide but haven't done yet, they are expressing TBR2. And so the red in here is showing you where in the brain these progenitor cells are. And then the presence of new neurons or other types of cells on the basal side, on the outer part, basically, of the brain. So the idea here is there's a couple of things that you've got these stem cells amplifying, and then there's this series of divisions that leads to the actual neurons and other cell types, and that the brain is a layered structure. So it's not just a tube anymore. You're building up these layers with the the neurons kind of towards the outside, stem cells during the development stage on the inside. I've included some terms here which I forgot to take away the underlining, but neurons are as you know, nerve cells that actually transmit nerve impulses, and then glia are support cells that sometimes look very much like nerve cells, but they're not actually transmitting impulses. They are support cells. So Schwann cells are glia of the peripheral nervous system. So these are the nerves that are not the brain and spinal cord, but that connect with the brain or spinal cord, either sensory input or motor output. Astrocytes, they are called astrocytes because they look kind of like stars. They have this kind of starry kind of appearance. And they are glia as well. They're in the central nervous system, so the brain and spinal cord, and they have to do with the regulation of nutrients and with controlling blood flow and so on. So very important for the nourishment and support of the brain. Oligodendrocytes are also CNS, brain and spinal cord, central nervous system, and they're involved with the myelination of neurons. So you probably know that there are myelin sheaths around certain types of nerve cells, and maybe that you may know also that there are medical conditions where myelination of neurons is lost, and they are very awful conditions in many cases. This is a kind of it's like an insulating wrap on a wire. They're important for rapid nervous transmission and so on. Of course, there's a lot more to it than that. I'm not a neurobiologist. So we're going to look just a little bit more at the development of the neural tube. And just sort of the symmetric versus the asymmetric cell divisions. And so the proliferation zone is this kind of area here. This is where this is the proliferation zone. So there's a few things to note. You've got a bunch of these neural stem cells, and what you can't see very well from this picture is that there's the cell body, which contains the nucleus, but there's this long process, and there's a connection to the most basal side and to the apical side. The cell bodies tend to be at the apical end, so closer to the ventricle or the interior of the tube. Now there are two proteins to know. There is Notch, which is the red, and there's Num, which is the blue. And just before cell division, these two proteins distribute unevenly in the cell. So on the apical side, you have most of the Num, and on the basal side, you have most of the Notch. Num is actually, its job is to inhibit Notch function. Okay, so when Num is present, it is preventing Notch from signaling and forming the neural fates, right? So some of the earliest cell divisions that happen are what we would call vertical cell division. So when the cells divide, both cells contain some Notch and some Num, and so that keeps them at the stem cell fate, and they remain stem cells. And so this is the symmetrical division, and this is the proliferative part, where you have lots of cell divisions to form lots of these stem cells. At some point, there is a trigger, and now the same thing happens. The cells have their two different proteins differently distributed in the cell, but this time the plane of cell division is horizontal, and this is asymmetrical because you're going to have a cell that is more apical, and it has pretty much all the num. So it stays as a stem cell, and in a little while it can start to express more notch as well, and it goes through this whole process again. But the other cell, the cell on the basal side, it has all the notch and it doesn't have the num, and it makes a connection to the basal surface. And this one is going to form either a neuron or a glia. So it's going to differentiate into one of those two types of cells. Okay, and it will stop dividing at this point, and it will just take on whatever its fate is. Now, the embryonic brain and nervous system over time produces many more nerve cells than are going to be needed. Some of these are eventually kind of pruned away, and I don't think we'll be talking about that in a later section. But so I'll just mention that. The fact that this stops dividing is okay because there's going to still be tons and tons of stem cells, and there's going to be more neurons than are really needed in the brain. Okay, and then this is just a little from this this paper, which just explains a little bit of things that have already been said, but just it gives you a better picture, because while all this stuff is taking place, the brain is also growing. So there are additional layers that are happening over time, and the brain is getting thicker and so on. And this is just a little bit of a schematic, and I've only kind of indicated the little bits that that you need to know. So this is gestational week. So this is a schematic of what human development would look like. Gestational week 6 to about, you know, a little over 30. Again, the ventricle, apical side, and then the basal side out here. And notice this growth. You're getting a thicker, bigger area. And I'm not, please don't try to memorize all the names of these layers. That's not the point. The point is just this shows you, these are the symmetrical divisions. They've got this symbol by them. And note just the connection to both the apical side and the basal side. But then the first asymmetric division that occurs, the more apical cell still looks the same, but the more basal one is going to form a neuron or a glia a little bit later in development. And this cell, rather than sort of magically making it to the basal side like on the previous picture, the fact is that it uses this process, and it kind of migrates up this process. And then when it gets up there, it starts to form final neurons. So these ones with the kind of triangle appearances, these are the final neurons. And the first ones that go up there are the kind of the lower layer. So they're at the lowest layer of where the functioning neurons are. And then the ones up here, these are sort of upper layer, right? Development has proceeded further. And so you can see these cells happening, these different kinds of, and this is another asymmetric cell division, and here's the cell making its way to this. It can spiral up and migrate its way up on a different cell as well. It doesn't have to always be the same cell, right? And so the key things to note are just that you're going to have these asymmetric divisions. The stem cells stay put, but they provide a scaffold, at least at the beginning stages, for the migration of the cells that are going to differentiate into neurons up to the more basal surface, right? And so the things to pay attention to are the ones I've circled, the migrating neurons. NEC just means the neuron stem cells. And this is what we mean when we talk about newborn neurons. We're not talking about newborn children. We're actually talking about when a cell after division, they're we say that the neuron is born, and so it's called a newborn neuron. It's just been developed. And then once it gets up here, that then differentiates into its final form. And I think that might be everything. I know there's a lot in this diagram, but I think I've pointed out the things that are most important for understanding. Again, I'm not looking for you to memorize this figure or everything about it, because there's all these different other things going on, but simply just the idea that the stem cells are providing, the scaffold for kind of migration of the neurons up, and that it's an asymmetric cell division that gives you one stem cell still, but then one cell that's going to differentiate. And that's a really common thing. Stem cell that divides, and it's going to form one that's differentiated, but one that's a stem cell, right? And then there's going to be another division, one differentiated and one stem cell. And that's why we call these ones self-renewing. And that's why this diagram shows like this, because it means this stem cell, one of the daughter cells, is exactly like the original stem cell. And that's a common way of describing that. Okay, I'm going to stop now because there's a lot in this lecture, and I would like to divide it into sections. We're going to look at the spinal cord next.